Okay, so chapter 6 and 1.3, part 2. We're going to start with proteins. Now, proteins are extremely important. This is the predominant organic molecule that you find in cells. In other words, after the water, okay, which is the majority of the makeup of the cell, proteins are next. Okay. Now, these are important for two reasons. One, they are structural components of the cell, and two, they are functional components of the cell, in other words, enzymes. Now, what this means is that virtually everything in the cell is made of or with proteins. So it's not made of a protein, and that's what we mean by structural components, being made of a protein. Chances are it was made with a protein. So in other words, an enzymatic reaction happened to actually make that particular component of the cell. Okay. Now, one of the most important things about proteins is that they have a very specific, unique shape. Okay? And that three-dimensional shape is extremely important because it dictates the function of the protein. Okay? You've heard this in classes before. We'll reiterate it here. Form dictates function. How the proteins are shaped is a very big deal. Now, if we go back to the monomers and polymers, one thing to remember, amino acids are the monomers of proteins. Other terminology that you'll hear in reference to proteins, anytime you hear peptide, amino acids are actually peptide bonded to one another. We call a short chain of amino acids a peptide, and a longer chain of amino acids a polypeptide, so usually more than 20. Okay. So polypeptides and peptides put together usually make up individual proteins. An enzyme is a special category of protein that is specifically meant to catalyze chemical reactions. I always tell people everywhere you see an arrow, there is an enzyme. So when you see formulas like this, okay, A and B together are used to make C. Everywhere you see that arrow, that means there is at least one enzyme working. So this is always a big clue. We're using enzymes. Okay? Antibodies themselves are actually glycoproteins, which is a lipid, which was the very end of the last chapter. I'm sorry, a sugar, which was the end of the last unit. Okay, so glycoproteins, sugars and proteins complex together. So when we talk about antibodies actually being used to attach to things like bacteria or viruses, those antibodies actually have a very important protein component to them. The attachment portion is a three-dimensional shape that specifies for an individual type of organism. So that three-dimensional shape, extremely important. Okay? It's so important that we actually spend a lot of time talking about protein structure if you study proteins. Now, when we talk about the primary structure of a protein, okay, this is basically that polypeptide chain. Okay? We're saying this is the amino acids connected to one another. Okay? Now, at this point in time, there is no function. Okay? A long strand of amino acids, it's just a string of things. It's not, doesn't have functionality. It doesn't have a working premise to it. Okay? But there are going to be chemical side chains that actually hang off of these amino acids and begin to do things like attract one another. Okay? So eventually the amino acids fold in on each other. Okay? This gives rise to what we consider secondary structure. Okay? And in the secondary structure, what we see are alpha helices, or an alpha helix, and beta pleated sheets that arise from which amino acids are in a specific order and which ones end up attaching to one another. Okay? So you can see that here. Here's your beta pleated sheet and your alpha helices. Okay? Helices is plural, so this would be an alpha helix. Okay? This is what we would consider a random coil. Okay? Not everything works with alpha helix or beta pleated sheet, but we do see sort of randomized arrangements. It depends on the order of the amino acids, so it depends on the protein you're making. Okay? When it comes to tertiary structure, now this is where we get three-dimensional. Yay! Okay? Because three-dimensional structures have functions. Right? Three-dimensional structures are working structures. So we see those alpha helices and beta pleated sheets begin to fold in on one another. Right? So this would be your tertiary structure. Okay. Now, some proteins stay at having a tertiary structure, and they're completely functional with just the tertiary structure. Okay. But there are categories of proteins that actually have to be formed from multiple polypeptide strands actually. So when it comes to quaternary structure, what we see here is multiple tertiary structures actually folding, okay, and then joining with one another. A good example of this is hemoglobin. Hemoglobin has quaternary structure because it is made of two alpha subunits, okay, so two alpha basically proteins, and two beta subunits, or two beta proteins. So it actually has four, okay, individual grouped proteins joined together. So hemoglobin is an example of having quaternary structure. Okay. Now, proteins that are in their correct three-dimensional shape and are functioning, ready-to-use proteins are in what we call the native state. Okay. Now, because we know that form dictates function, what that means is when we change the form, okay, the function is lost. Okay. If you take away the form, you take away the function. When protein structures are changed or disrupted, we term that being denatured. So denatured proteins, what it means is that we changed how these structures are put together. Okay? We broke them apart in some way, and if that happens, they will no longer work. Okay? So, this is sort of the very basics of nucleic acids. I'm not going to hit it particularly hard. One of the reasons is because in the next unit, so for the third test, we're going to spend a lot of time on genetics. So I'm going to get a lot more into this, okay, before the next test. Now, that is test 3. So when it comes to the nucleic acids, okay, remember our monomer here, okay, is the nucleotide. Okay. And a nucleotide is actually made up of 3 individual parts, a phosphate group, okay, a sugar that can be ribose or deoxyribose depending upon the nucleic acid you're trying to make, and a nitrogenous base. Okay? So all nucleotides will contain a phosphate, a sugar, and a nitrogenous base. If you think about those chunks that we were talking about earlier in the chapter, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, well, the phosphate group is going to have phosphorus and oxygen, the sugar, carbon, hydrogen, and oxygen, and the nitrogenous base, nitrogen's obviously found in there. So we're actually utilizing pretty much every piece of this, with the exception of the sulfur, which honestly those are mainly found in things like amino acids. Okay, so these are important components of individual nucleotides and therefore DNA and RNA. Now, the importance of DNA and RNA lie in this, and this is one you definitely want to remember. Okay, this central dogma of biology, or central dogma of genetics, and it is that DNA, okay, will be used to produce RNA, and RNA will be used to produce proteins. Okay, so the DNA being used to produce RNA and the RNA being used to produce proteins, turning DNA into RNA, or using DNA to produce RNA, excuse me, is termed transcription. Okay. Using RNA to produce proteins is termed translation. Okay. But the big thing here is proteins. Okay. If you remember, the proteins were making up pretty much everything within the cell. Okay. Or at least had some part in making up everything within the cell. And because they're enzymes, they're also going to deal with the everyday metabolism of the cell. In other words, your DNA is your instructions on how to run a cell. It's on how to build a cell, how to make a cell work. That's the thing that makes your DNA so important. That and the fact that we actually pass it on. It's heritable information that we're moving from one organism to the next, from parent to offspring, or from mother cell to daughter cell. Okay? The RNA is actually what we consider, well, your book considers it a helper molecule. The reality is you cannot make proteins without RNA. DNA is sort of your go-between. Okay? It helps rewrite the DNA into a usable format so that that usable RNA can then be turned into proteins. Okay? Now, the last of the nucleic acids I'm going to talk about here, ATP. Okay? ATP is itself built around a nucleic acid. It's basically a nucleotide that instead of having one phosphate group actually has three attached to it. If you're unaware, ATP stands for adenosine triphosphate. Okay? Now, this is important if we all remember because this is a high energy compound. I always tell people this is a temporary energy storage molecule. And it's important because ATP actually allows us to have usable energy. It's going to let other places within the cell actually be able to utilize energy. And it's another example of a recycled molecule. We use ATP by breaking off that third phosphate, okay, and moving the conversion of ADP, or ATP, I'm sorry, to ADP. And then we recycle this, we bring it back by converting ADP to ATP, okay, by continuously chopping off phosphates and adding new ones. Okay? The issue is we've got to have energy to add on new phosphates, so our energy from our food is used to add new phosphates onto ATP so that we can utilize it, okay, somewhere else within the cell or within the body. Let's look a little bit more closely at our ATP molecule. Okay? Adenosine, okay, this is a sugar, specifically it's a ribose sugar, okay? And this is one of our nitrogenous bases. Okay. Specifically, it is an adenine nitrogenous base. Okay. So an adenine and a ribose sugar are what help make the adenosine part. Okay. Now, if you remember when we talked about lipids and we talked about phospholipids, these phosphates are large charged molecules. Okay. So these large charged phosphates, we're actually going to try to butt them up against one another and hold them together. The problem is they all have the same charge, so their natural tendency is to repel. Okay? Now, because of that, okay, holding these phosphates together, okay, takes a high energy bond. Okay? And holding the first two together, okay, and on to that large adenosine, okay, these don't have anywhere near as much energy, okay, as we would see, okay, with this guy right here. Okay? This is your high energy storage bond. Okay? This is where all of your energy is. And if you remember your chemistry, okay, breaking bonds releases energy. So when we break bonds, we end up releasing energy. So breaking that bond releases enough energy to be used. Right? And it's going to take energy to remake the bond, but we're actually going to snatch that energy off of another molecule, basically, for example, glucose. As we break down glucose in cellular respiration, we're going to take that energy and use it to replace phosphates. Okay? So we're going to constantly recycle from ADP to ATP, adenosine diphosphate to adenosine triphosphate. And then we're going to use that phosphate, we're going to use that energy, okay, elsewhere within the cell. When I say temporary energy storage molecule, I mean temporary. Okay? ATP is used almost as quickly as it is made. Okay? So, This moves us back into chapter 6. Let's talk a little bit about nutritional categories of microorganisms. Okay? We base organisms and we begin to categorize them based upon what types of energy sources that they use. Now, when we talk about energy sources, the reality is we're not just talking about energy. We're talking about what they use, okay, to begin actually grabbing energy. And there are two options here for energy sources. Okay? Sunlight, okay, and chemicals. Okay? When we talk about chemicals, what we mean is breaking bonds. So breaking bonds in molecules. Okay? To go along with this, we take into consideration carbon sources. Okay? Because this is going to be an extremely important nutrient, since pretty much all of your macromolecules are going to be built around carbon. It's a necessity. Okay? We see inorganic carbon sources and organic carbon sources. Okay. Now, when it comes to putting these together, okay, one thing you'll see common on the end of all of these terms is the word troph. Okay? To be trophic, it's basically feeder, okay, or eater. Okay, so an organism that uses sunlight as an energy source will be called a phototroph. Okay, an organism that uses chemical as an energy source will be a chemotroph. See, we didn't name them that difficultly. Okay, now organisms that use inorganic substances, okay, are basically going to be creating their own organics. Okay, so what they're going to do is take those inorganics and convert them to organic substances. Okay, they make their own organics. Okay, so we call them autotrophs. Okay, and then everyone else that uses organic molecules, ready-made organic molecules, will be termed a heterotroph, because hetero means other. So if you can't make them on your own and you're not an autotroph, you are a heterotroph. Okay? Examples of this, photoautotrophs, like plants, go through photosynthesis, where they use sunlight and carbon dioxide to produce their own sugars. Okay? Glucose is a product of photosynthesis. So sunlight and carbon dioxide, which we remember is an inorganic molecule, okay, will be photoautotrophs. Meanwhile, a chemoautotroph does not use sunlight. It will use some type of chemical in an inorganic compound, okay, to actually create its own organic carbons. Okay, this we see pretty rarely. Certain bacteria, things that you'll find in places where there's a lot of heat or a lot of ability to utilize chemical energy. Methanogens is the example that's given here. These basically metabolize methane. Okay? Meanwhile, a photoheterotroph will use sunlight and organic nutrients. This one's also somewhat foreign. Okay? Purple and green photosynthesizing bacteria. These do not really use carbon dioxide the way that plants do or the way that algae does. Okay? The chemoheterotrophs, okay, basically what they're going to do is use their organic compounds as chemical energy and as their carbon sources. Okay? This is us. This is where we're fitting in. Most of the bacteria are going to fit in here. Fungi and protozoa, we see lots of large multicellular non-photosynthesizing organisms fall into the category of being a chemoheterotroph. Okay? To be honest, pathogens are going to fall into this category. They're utilizing us as food, so they're going to utilize the same food sources that we do. Okay? A few other terms that are commonly used: saprobe, which I have used before. Okay? If you remember, I liked the term saprophyte. Okay? These are organisms that live off of decaying material. Okay? If you remember, sapro means to rot. Okay? And then the parasites, which technically are actually normally chemoheterotrophs, they just happen to be living off of tissues or fluids. Okay? very specifically. So those are a subclass of chemoheterotroph. Let's talk about the parasites a little bit more. Again, deriving their nutrients from the cells and tissues of a living host. Okay. The pathogenic parasites will cause damage as they derive their nutrients from their living hosts. Viruses, helminths, protozoa, we know lots of different types of pathogens. Okay. What we term ectoparasites, which live outside of the body. Okay. Think mosquitoes, leeches, okay. Things like a superficial fungal infection would be ectoparasite. Okay. Endoparasites live within their host. Okay. These would be the helminths, lots of the bacteria, some of the protozoa. Okay. There are also what we term intracellular parasites. Okay. These are actually living inside of a cell. Okay. Viruses are a great category of these. All of your viruses are intracellular pathogens. Okay. Because their entire goal in life is to hijack a host and live inside of that. Well, not necessarily live, but reproduce inside of that host cell. Okay. Leprosy, the bacillus that causes leprosy, lives inside of things like macrophages. You could say the same thing for TB. The spirochete that causes syphilis can be intracellular. Okay. We have obligate parasites. Now, this is a new term. Okay. Anytime you see this word, obligate, okay, it is necessary. It needs it. Okay. So an obligate parasite needs to be a parasite. It has to have a host. Now, organisms that are less strict than being an obligate parasite, we can culture artificially, like the ones we have in the lab. These are obviously not obligate parasites because we're not providing them with a host to survive. Okay? They don't need to be inside of another cell or inside of a living organism in order to survive. Now, again, okay, let me emphasize this one more time. The majority of microorganisms that cause human disease are going to be chemoheterotrophs. They're living off of our nutrients. Okay. Okay. So our okay, carbon sources. Okay. Oh, I don't know why that keeps happening. Okay. Are going to be the organics. Okay. Their energy sources are probably going to be the same thing. Okay. Think sugars. Okay. So that's our case for the chemoheterotrophs. Okay. So since we've talked about nutritional requirements, let's talk a little bit about how microorganisms actually get these nutrients inside. Okay. You can't have one without the other since a lot of these are going to be unicellular. We're going to be talking a lot about passing things across that cell membrane. Because remember, the cell membrane's job is to regulate what comes into and out of the cell. One of the things we're definitely going to want in the cell is going to be nutrition. Okay? We're going to want those nutrients on the inside. So, we've got to get nutrients past the cell membrane. We have to get them past the cell wall. That's usually not an issue. The cell wall doesn't do a whole lot of regulation. Now, the driving force for transport for all organisms is atomic and molecular movement. So basically, particles that naturally are going to repel one another and push off of each other and move around. Okay? Now, our microorganisms are mainly going to be living in very aqueous environments, so there's going to be a lot of water. Makes sense because that is one of the components that they're going to need most inside of the cell. If you remember, the cell is 70% water. So what we're dealing with is bringing things into the cell that are floating around in the external environment. Okay? So probably one of the easiest examples of this is diffusion. Okay? Now, we're going to start off with the passive transport mechanisms, and diffusion is a great example of a passive transport mechanism. Okay. The issue here when we talk about passive transport is that there is no ATP expenditure. Okay. There's no ATP used. This is just normal Mother Nature molecular movement. Uh, Honestly, this is based completely upon the fact that things move towards equilibrium, and it's true for every type of environment. Okay. So remember that we move toward equilibrium, and it happens naturally, okay, with no ATP needed. Okay. Now what that means is that there's more of something on one side, okay, of an environment. It will naturally spread its way out to other places within the environment until it reaches equilibrium. Okay? So we see things in diffusion move from areas of high concentration, like right here, okay, towards the areas of low concentration until they reach equilibrium. Uh, Osmosis is a great example of this. It's actually normally considered the diffusion of water through a selectively or differentially permeable membrane. Being selectively or differentially permeable means that the membrane will allow some things to move through easily, while other things are not allowed to move through easily. Uh, the gist here is that for most organisms, they have set up their cell membrane so that water can constantly move in and out of the cell, because lots of times with it, the water will bring nutrients. Okay? That's true for our cells. We have things like aquaporins that, even though there is that hydrophobic layer between the external environment and the internal environment, okay, that water is allowed to move freely from areas of high concentration to low concentration. Okay. You can see it here. This is an example. What we're seeing is a water molecules. They're saying all these little blue guys right here are water molecules. Okay. Uh, Now, the issue is that because there is a solute, something like salt or sugar, there's something dissolved in the water within this sac. The sac itself is going to be considered a semi-permeable membrane. Look here, this is your semi-permeable membrane. What you can see is it's semi-permeable, or differentially permeable, really means the same thing, is that these molecules are too large to move through the pores in the membrane, but the water molecules are not. Okay, so the water moves freely. Okay, and that solute that is dissolved in the water is stuck. Okay. Now, because of that, that solute is taking up space. Okay. So if you look out here, this has lots of water. It's pure water. Okay. So there is a high concentration of water, high concentration of H2O. Okay. Meanwhile, inside, because that solute is taking up space, okay, the concentration of water, it can't be 100% on the inside. There's solute in there, so it's at 99% or 95% or 80%. Okay, so this is a lower concentration of water because of the presence of the solute. So the water is actually going to begin to move, okay, into this particular system. Okay, we call these fault cells sometimes. This little fault cell system with the differentially permeable membrane. The water is going to continuously move in. You can see it actually rising here. Here's our water line on this one, the water line, the water line. Okay, more water will continuously move into the synthetic cell because, okay, it's trying to reach equilibrium. We're trying to get the same amount of water in the bag as we're trying to get outside of the bag. It's all about moving towards equilibrium. Okay. Now, most cells are going to be surrounded by some free water, okay? And that water moving in and out is going to have a major impact on cellular activities. I've said before that water is the basis of reactions within the cell. So if cells end up missing water, okay, then one thing that we see is that they stop running important chemical reactions or don't have the ability to run important chemical reactions. Now, one thing that's brought up, and I know this is one of everybody's favorite subjects, is that when we start talking about water moving into and out of cells, we talk about the relationship between cells and the environment, and that's something we term tonicity. Okay? This has to do with relative concentrations of solutes on either side of the membrane. So, tonicity is a comparative term. We're comparing two things. Okay? You can compare one solution to another. You can compare the inside of the cell to the outside of the cell, or the outside of the cell to the inside of the cell. But the wording matters. Okay? The wording that is used is important. Okay? So, in an isotonic situation, what we see is equal amounts of solutes on either side of this membrane. Okay? Inside of the cell and outside of the cell, or between the two liquids. Okay? So, isotonic, we see equal amounts. Okay? When you use the term hypotonic, you're saying that one solution has less solute than another. When you use the term hypertonic, you're saying that a solution has more solute than another. Notice that tonicity does not reference the water. Tonicity is about the solute. It's about the stuff dissolved in the water. The sugar, the salt, the protein, you name it. Okay? It's about what you find within the water. Okay? Not the water itself. You want to talk about water, you got to talk about osmolarity, and that's a whole nother subject. Okay. So, let's throw some examples out there. Okay. If solution A is blank to solution B, if solution A is isotonic to solution B, what will that mean about solution A? Any guesses? Try to write it down. I'm going to give you a second. Hopefully you got in there that means that A has the same concentration of solute as B. Okay. And that B has the same concentration of solute as A. Remember, isotonic means we're dealing with equal solute concentrations. Okay. If A is hypertonic to B, okay. Try that one. Well, what we see is that A will have a higher concentration of solutes than B, and what that means is that B will have a lower concentration of solutes in solution than A. How about the last one, hypotonic? If A is hypotonic to B, hopefully you're getting the hang of this. What we'll see is that A has a lower concentration of solute than solution B does, and that B has a higher concentrations of solutes in solution than A. Okay? It's all about the wording. I could have just as easily asked B in reference to A. Okay? So make sure you remember, read the words carefully. If all else fails, draw yourself a picture. Fake it. Okay? Draw yourself a cell. Here we see a cell in isotonic solution. Fake numbers if you need to. Say this is 20% sugar on the outside and that it's 20% sugar on the inside. Okay, so we have equal concentrations of solutes. Okay, now one thing that people have a tendency to forget, okay, is that lots of times when you get asked questions about this, they don't ask you about the solute moving because it's a semi-permeable membrane. The solute stays put. We like to ask about where the water goes. Well, if it's 20% salt, that means it's 80% water on the inside. If it's 20% sugar on the outside, that means it's 80% water on the outside as well. Okay. So is there going to be any net movement? Okay. Well, here's a hint. Water always moves. Water is continuously moving into and out of the cell. But will we suddenly have a lot of water move one way? No. because this is a system that is already at equilibrium. We're good. Okay. So, what about cells in hypotonic or hypertonic solutions? Okay. So that solution on the outside is hypotonic. So we'll say there's 10% salt on the outside and then 30% salt on the inside on this one. Okay. Or if we go over here in the hypertonic solution, what if we say that there's 30% salt on the outside and this one only has 10% salt on the inside of the cell? Okay. Well, that doesn't give you all of the information, especially if I want to know about what happens to the water. So what do? You go in and say, all right, well, that means this one is 90% water on the outside, and this one is 70% water on the inside. The only thing that can move is the water. The salt won't move in and out. So we want to see things move towards equilibrium. Where am I going to go, high to low? That means water from the outside, where it's 90%, will begin to move inside, where it's 70%. So that cell will probably swell, and it's a possibility that it could lyse. Now, remember, lysis doesn't happen in bacterial cells because they have the extra protection of the cell wall. Cell walls prevent lysis under osmotic pressure. Okay? So when the water rushes in, these cells don't burst. Okay? So if we move over here to our cell in hypertonic solution, okay, that means if it's 30% salt, the outside is 70% water. And 10% salt on the inside means that this is 90% water. Okay? What way will the water move? Well, it's going to move out because the water is attempting, okay, to bring these cells into equilibrium. Okay? So as this loses water, that salt concentration goes up. As this gains water, the salt concentration goes down. We're moving towards equilibrium. Okay? Draw yourself a picture. Okay? All right. So enough, okay, about tonicity. I'm going to stop right here. We are going to move into active transport. Actually, okay. So enough about tonicity. Let's talk a little bit about other types of transport. We've already been over simple diffusion. Basically where there is no barrier between things moving in and out of the cell. They move freely at will and towards equilibrium. We do sometimes see facilitated diffusion. Now, this is still passive diffusion. There is no energy requirement. We're moving from high to low. The difference is that facilitated diffusion is moving molecules that won't pass easily across the membrane. Okay? So in facilitated diffusion, we need help. That's what it means to facilitate something. Okay? So this is helped diffusion. Okay? It's still diffusion. The difference is here we're going to use a helper molecule. There's going to be something, probably a protein, okay? A transport protein that's going to help move molecules from high to low because we're still working on that Mother Nature likes things at equilibrium. Okay? This time, we just don't need to expend any energy, but we do need a helper molecule. Okay? Now, active transport is what's coming up next. It is the opposite of passive transport. The exact opposite. We are going to move things against the concentration gradient, and that's going to take ATP. We're trying to shove things back up the hill or swim them back upstream. We're going from areas of low concentration to high concentration. Okay? In other words, we're shoving in more. That's not what Mother Nature likes. Mother Nature wants everything spread out. So let's talk a little bit about active transport. Okay? This is a nice way to remember it. And you see I color-coded it here. All the A's go together. Active transport, cells expending energy from ATP, okay, enabling them to move substances against the concentration gradient from low to high. You're shoving extra things into that junk drawer. They don't want to go. It's going to take more energy. Okay. Common substances transported actively, monosaccharides, so sugars, Okay? Amino acids, organic acids, phosphates, metal ions, that's a lot of things that the cell is very commonly going to need. Okay? Because of that, what we see is that active transport is the most common type of transport that occurs within a cell. Okay? That a lot of things are going to be actively brought into the cell. Think about it, food, even if there's not a lot of sugars around, the cell is going to want them. Okay? So even if there aren't that many sugars in the external environment and it can't just move them from high to low, it's still going to want them in the cell. So it will expend the energy in order to bring in, okay, 1 ATP or 2 ATP used to bring in a glucose that's going to get you 30 plus ATP. It's worth it. Okay? Actively transport those things in. Okay? Other types of active transport we see, endocytosis. Okay? Yes, this is a type of active transport, okay, where cells actually engulf things external to them. Okay? Phagocytosis, where we move in solids. This is endocytosis of solids. Okay? Or pinocytosis, which is basically an endocytosis of liquids, moving things in like oils. Okay? Water does not count when it comes to pinocytosis. We're usually moving in things like pockets of oils or pockets of fats. Those are sort of the most commonly pinocytosed things. Lots of times we refer to this as cell eating for phagocytosis and cell drinking for pinocytosis. Okay. All right. So I'm going to end part 2 here, and when we come back for part 3, we're going to talk about environmental factors that influence microorganisms. Okay? Be prepared. We're going to talk about all of the different types of external environmental factors that can influence how the organisms grow and how well they function. Okay? This is going to be a lot about survivability.